So now I'd like to spend a little bit of time talking about a very important concept, and that concept is that of respiratory control. Respiratory control is really how our body responds to the environmental needs that it places itself in, whether we're exercising, whether we're resting, etc. So when respiratory control exists, that's most of the time, in fact 99.99% of the time, our body has respiratory control going on. Usually we have to disrupt it with something before we can have a loss of respiratory control, and that's usually pretty drastic circumstances. So let's consider first of all what's happening in respiratory control. In respiratory control, the most important concept to remember is that electron transport is directly tied to oxidative phosphorylation. And oxidative phosphorylation is directly tied to electron transport. That goes both ways, meaning that they are interdependent upon each other. If I stop electron transport when I have respiratory control, I will stop oxidative phosphorylation. And similarly, if I stop oxidative phosphorylation, I will stop electron transport. So this intertwining of these two is a very important concept. And with this, we can learn a lot about what's going on. So let's imagine, first of all, that we're sitting around the house, we're not doing anything, we're not exercising, we're resting. What circumstances do we have? Well, first of all, when we're doing that, we're not using our muscles, and our muscles are one of the major sources of burning ATP. When we're not using ATP, our body catches up, makes ATP, and our ATP levels end up being high. When ATP levels are high, that means that ADP levels are low. Well, why is that important? It's important because we have to remember that ADP is necessary for the process of oxidative phosphorylation. That ATP synthase that I talked about earlier that rotates requires ADP. That rotation of the ATP synthase makes ATP. It also allows protons to come back into the mitochondrial inner matrix. So if there is no ADP, oxidative phosphorylation can't occur. And if oxidative phosphorylation can't occur, no more ATP will be made because, of course, there's plenty of ATP to begin with. But more importantly, protons can't move back into the matrix. Well, what electron transport is doing all the time is pumping protons out of the matrix into the intermembrane space. And the more those protons get pumped out there and don't make it back in, the higher the gradient. That is, you'll have more protons at growing amount because they're not coming back in, and that makes it harder and harder for the electron transport system to continue to pump protons out. Well, eventually what happens is the electron transport system can no longer push protons out. And as a consequence, the gradient of the protons is so high that the charge difference across the membrane is so high that electron transport itself will stop. So we can see here that the resting situation stops oxidative phosphorylation and subsequently stops electron transport. When electron transport stops, what happens? Well, what happens is that the NADH and the FADH2, which had been dumping their electrons into the electron transport system, now have no place to put them, because when the electron transport system stops, the electrons stop being transferred, which means complex 1 is full of electrons, no place to put them. Complex 2 is full of electrons, no place to put them. All the complexes are full of electrons because they can't move any further. That means, of course, that since NADH is accumulating and FADH2 is accumulating, then what the citric acid cycle, which requires NAD and FAD, no longer itself is able to function. So the upshot of all this is that when we run out of ADP, then oxidative phosphorylation stops, electron transport stops, and citric acid cycle will stop. Glycolysis, which also needs NAD, may also stop, but there's a little bit more of a consideration for that. Now, let's imagine another situation where instead of resting, I'm out and I'm exercising. So if I'm exercising, what's happening? When I'm exercising, my muscles are using ATP and making ADP. Well, if I've been sitting around this whole time and I had everything stopped as I've just described to you, what I'm now describing to you is a circumstance where everything is going to start, okay? Because when I start exercising, what happens is the ATP gets converted into ADP. You remember that ADP was the limiting nutrient that was necessary for oxidative phosphorylation to occur. So ADP now becomes available. What happens? Well, the little turbine that spins that makes the ATP, excuse me, the turbine that spins that makes the ATP can now start spinning. And as it starts spinning, the protons, which were now in high concentration outside of the mitochondrial inner membrane, okay, now start flowing through the ATP synthase. Consequently, the proton gradient starts falling. And when the proton gradient starts falling, electron transport is now able to start pumping protons again, because that is the limiting factor when there's no ADP present. So the electron transport starts moving electrons, starts pumping protons, and the moving of electrons now empties the electrons out of complex 1 and complex 2, allowing them to take up NADH and FADH2. Consequently, the citric acid cycle starts. Now, because of all these things, we learn that exercise allows us to burn things. Citric acid cycle is an oxidative pathway that is literally burning things. Exercise is going to help us to use more resources, and using those resources will help us to lose weight. On the other hand, if we're sitting around and we're not running oxidative phosphorylation and we're not running electron transport, we're not using resources, we're going to gain weight. So when we have respiratory control going on, we can see how electron transport and oxidative phosphorylation are tightly linked. And that linking happens because when respiratory control exists, the only way that protons can make it back into the mitochondrial inner matrix is through the ATP synthase complex. All right? So compounds that disrupt that is, compounds that allow the protons to bypass the ATP synthase and get back into the matrix in another means, do what we call uncoupling of electron transport and oxidative phosphorylation. And that uncoupling also destroys respiratory control. What you see on the screen is a molecule called 2,4-dinitrophenol. And 2,4-dinitrophenol has the property that I just described to you. 2,4-dinitrophenol will insert itself in the membrane, the inner mitochondrial membrane, and permeabilize it to protons. Now, 2,4-dinitrophenol, as I mentioned in class, is not a natural compound. It was marketed back in the early 1900s as a diet drug, and it killed a lot of people. It did, in fact, cause a lot of things to happen because of the uncoupling that I'll talk about in just a minute. But nonetheless, it was a very dangerous compound. Our body does make some natural uncouplers. I'm not going to talk about them here, but they do relate to something that our body has called brown fat. And if you're curious about that, I would recommend reading up on brown fat. But 2,4-dinitrophenol is an uncoupler, and it has this property of burning resources because of the uncoupling. So let's talk a little bit about what happens with that. When 2,4-dinitrophenol is added to cells, it will get into the cells and it will go to the mitochondrial inner membrane, where it will permeabilize the membrane to protons. Now, you remember that when we had tight coupling, what happened was the protons had to go through the ATP synthase. With 2,4-dinitrophenol in the membrane, what happens is the protons go directly through and they don't pass through through the ATP synthase. It's like playing Monopoly and you don't go and you don't collect $200. Well, the $200 you get in Monopoly is the ATP that you would get if the protons went through the ATP synthase. You're bypassing the synthesis of ATP and you're destroying the proton gradient at the same time. Well, let's think about the effect that this has on the cells. The limitation when you have respiratory control is that the oxidative phosphorylation, when it stops, will stop the electron transport. And when the oxidative phosphorylation starts, electron transport will start. Well, what if nothing stops electron transport? That's actually what's happening with an uncoupling molecule like this. Let's think about review again the electron transport in the tight coupling, okay? So if I have no ADP, if I stop protons moving through the ATP synthase, when I have tight coupling, the proton gradient will grow when I'm resting. And as it grows, it slows down and eventually stops electron transport. That doesn't happen when 2,4-dinitrophenol is present, because no matter how much the electron transport system is running, the protons get pumped out and they automatically come back in, but they don't come in through the ATP synthase. Consequently, no ATP is being made, and cells, of course, need ATP. Well, meanwhile, electron transport is running like crazy. As electron transport is running like crazy, protons get pumped out, but they come right back in. NADH is readily being converted into NAD. FADH2 is readily being converted into FAD. And consequently, the citric acid cycle starts running like crazy because an abundance of NAD and FAD keep that pathway going. So we can see how this compound 2,4-dinitrophenol will facilitate the burning of nutrients without any exercise. That's why it was considered the perfect diet drug. The problem was it was killing people simply because they were not making ATP. Okay? So that's what happens when respiratory control is lost with an uncoupling reagent such as 2,4-dinitrophenol.